Across living Earth, proteins are built, folded, eaten, and returned. They fill grain kernels, milk, seeds, muscle, skin, feathers, and leaves. Each protein is a necklace of amino acids, clasped by resilient bonds. Those bonds give bread structure, hide strength, and seeds their quiet reserves. Yet abundance becomes a problem when proteins must be softened or reused. Heat can force them apart, but often bruises flavor, color, and texture. Strong alkali can break them, but leave salt, odor, and burden. Nature found a gentler key long before human workshops existed. In soil, bacteria harvest fallen cells with invisible protein-cutting tools. In ripening fruit, microbes nibble old tissues into new nourishment. In animal stomachs, proteases turn meals into building blocks. Among these cutters, one family favors the middle, neither acid nor alkaline. We call them neutral proteases because they work near water's calm center. Neutral does not mean weak, it means suited to ordinary living water. Their story begins with digestion, the first chemistry everybody feels. In the early 1800s, scientists watched stomach juices dissolve meat outside bodies. The sight was startling. Life's chemistry continued inside a glass vessel. In 1836, Theodor Schwann named Pepsin from the stomach's dissolving power. Pepsin was acidic, but it opened the imagination to biological catalysts. Later, Wilhelm Kühne studied pancreatic digestion and gave enzymes a common name. Trypsin worked in a milder fluid, closer to the body's neutral streams. But the countryside held another clue, quietly sitting in gelatin dishes. During the late 1800s, bacteriologists noticed some microbes liquefied solid gelatin. On benches, clear halos spread around colonies where gelatin had disappeared. The gel did not melt from warmth, it vanished by chemical touch. Italian investigator Claudio Fermi carefully linked this liquefaction to bacterial proteases. He showed that microbes could release protein-digesting power beyond their own cells. For neutral protease, that idea was decisive. Enzymes could be harvested. The great industrial shift came with 20th-century fermentation. Clear broths replaced animal organs as steadier sources of useful enzymes. Cultivated microbes could be fed, listened to, and selected with care. Bacillus species became especially important, sturdy soil bacteria with generous secretions. Some bacillus enzymes preferred alkaline conditions, others favored the neutral middle. Researchers named bacterial neutral proteinases, then traced their dependence on metal. A tiny zinc atom often sat at the heart of the cut. In Japan, hot spring soil yielded a remarkable bacillus enzyme, thermolysin. Isolated in the early 1960s, it became a model neutral protease. Thermolysin's structure later showed how metal, water, and protein meet precisely. Industrial neutral protease did not arrive as a loud revolution. It entered vats, tanneries, kitchens, and mills as a quieter alternative. Its strength was restraint, working near neutral conditions, sparing surrounding materials. Imagine a folded protein as a rope tucked into a pocket. Neutral protease settles against it, feeling for a vulnerable link. A water molecule is guided into place beside the bond. The metal center steadies the moment, like a hand holding scissors. One connection opens, and the long protein becomes two shorter pieces. The enzyme lets go, unchanged, and searches for another hidden seam. Nothing is smashed. The work is selective, repetitive, and remarkably quiet. This quietness matters in foods, where texture can be more valuable than speed. In soy and grain processing, neutral protease helps unlock savory peptides. In dairy and cheese preparations, it can shape softness and ripening character. In baking, it relaxes stubborn dough when flour proteins hold too tightly. In brewing, it helps reduce protein haze that clouds the finished drink. In meat and seafood, it tenderizes fibers without turning them to ruin. Beyond the table, leather makers use proteases to soften hides gently. Neutral action can reduce harsher chemical steps during baiting and cleaning. In textile work, protein stains and fibers can be treated with care. In detergents, proteases meet egg, milk, blood, and grass proteins. Neutral protease also helps transform surplus protein into soluble nourishment. Plant meals, fish trimmings, and byproducts can become more usable streams. In waste treatment, protein-rich residues can be opened before biological recycling. Instead of disposal, old protein can re-enter the economy as value. Each application rests on the same old discovery from soil and digestion. Life already knew how to unbuild proteins gently, one bond at a time. What changed was our patience to borrow the tool without exhausting nature. Modern enzyme work continues to favor mild water, moderate heat, and fewer wastes. Neutral protease reminds industry that strength can come from conditions near balance. Its past runs from stomach flasks to soil bacteria and fermentation vessels. Its future may run through cleaner food, better materials, and circular resources. In the middle path, biology offers chemistry a greener way forward. Behind Enzymes Bio is a team of scientists who love this quiet biology. We gather enzymes from across the world so researchers and businesses can order from one home. Nature's technology for a greener tomorrow. See more at enzymes.bio.